Good afternoon, children. We'll continue with our lesson on transpiration. When last time I had introduced with various experiments of the topic about the polythene bag, the use of cobalt chloride paper in an experiment like this with the bell jar. Then we had started with the measurement of transpiration. But last time, children, we had discussed only one method, that was the weighing method. Today we are going to start with the second method of measurement, which we call as Potometer method. We had also discussed last time various improvisations of the weighing method for measuring transpiration by the use of this graduated sight bottle tube. Then this another experiment for weighing method, right? Before we move further, let me remind you, children, of this diagram. In an average plant, I had told you, if suppose the amount is 100 liters of water being absorbed in a day by a large tree, then around 98 liters are being lost by transpiration. Some only 2% is being kept. Indirectly, the measurement method by potometer, we will not measure this water lost directly. That is, this water lost will not be measured, but we will measure this uptake. Definitely, we can say more or less this quantity is almost equal to this. So today, when we are continuing with our potometer, always remember, children, this will not measure the amount of water given out, but will absorb the amount taken in. On the very note, let us start with our new topic, potometer. Poton means to drink and meter means to measure. Potometer, by definition, it is a device that measures the rate of water intake by a plant, and this water intake is almost equal to the water lost through transpiration. Just now I showed you in the diagram that how this 100 liters which is taken, suppose in a hypothetical way, is almost equal to the water that is lost, around 98. So in the method we are introducing today, remember, we are going to measure the intake and not the giving out of water directly. So various types of potometer that we have. First is the farmer's potometer, which measures water intake. Just now I told you. Genong's potometer, this is the blue colored because this is in your course, children. The rest of the three, they are not. So Genong's potometer, it also measures the water intake. Darwin's potometer, which demonstrates the suction force created due to transpiration. Suction means a kind of pull, children, that is on the water column by the roots. Then Garrow's potometer, which demonstrates unequal transpiration from two sides of a dorsiventral leaf, right? So I'll now introduce two words to you, dorsiventral and isobilateral. Dorsiventral and isobilateral leaf. Both are types of leaves, children. The first is, this leaf has a dorsal and a ventral surface. In other words, this leaf is the one that we see with this type of venation. One is a main midrib and the other are side veins. We see such leaves in dicot plants. Alright, the upper side is darker in color and the lower side is lighter in color, right? And there's a clear difference between the trapping of sunlight. So always pick up a leaf like this anywhere around your home. You will always find with this venation what we call as reticulate. Reticulate means like a network, right? Such leaves are called dorsiventral and they are characteristics of dicot plants, or the plants which grow from seeds having two cotyledons. Just another category, we have isobilateral. Isobilateral, iso means same, right? Such leaves, they are aligned to such an angle that they trap equal sunlight. Iso means the trapping of sunlight will be equal from both the sides, right? And one more way to distinguish them is the venation, which is parallel. So such leaves you will always find in monocot plants, example, grasses. So if you pick up the grasses, you will see a very major difference. This was the dicot plant with the reticulate venation, and this is the parallel venation on monocots. So we are going to start with as I just mentioned this word, here, dorsiventral. Such leaves are called as dorsiventral, whereas the other category with the grass is called isobilateral. We'll talk about later about this in detail. Coming to Genong's potometer. Genong's potometer, it's a device which is in your course, as just now I told you, out of the 4 types. And here, before we start imagining the working of this, remember children, we are not going to measure the water lost, but we are going to measure the water uptake. Though we can use any twig for our experiment, but we are preferably taking a coleus leaf into this setup. How we make up? A twig of the plant is cut with a sharp knife, and there are many precautions to be kept at every step, children. Number 1, this twig that you are going to cut for the experiment should be cut obliquely. Why oblique? Now, okay, this is a stem if you cut it like this, or this is a stem if you cut it like this. Where do you have more surface area? This, transverse, or oblique? Guess it? Yes. This is the area oblique, which is more in surface area. So the twig that you are going to use here should be cut obliquely. This is number 1 precaution. Second, it should be cut under water, right? So that it should not absorb any air bubble. Alright, so twig is cut with 2 precautions in mind. Then the entire setup, children, is filled with water so that no air space is present. And now only 1 air bubble, remember, only 1 air bubble is to be introduced in this horizontal graduated tube. Graduated means it has a scale which is showing readings. Alright, this is done. Now how to introduce this air bubble? Remember, this is dipping into a beaker containing colored water. So this is just very instantly that we just lift up the capillary tube and dip it immediately, maybe for just a fraction of second, and bubble of air is introduced. Alright, so this is like by practice, though at times many bubbles come in into 1 second, fraction of second, you will see 4, 5, 6 bubbles. So how to shift them back? We don't need many, we need only 1 bubble. Then we require this reservoir. You can see there's a stopcock over here like a tap. So if we turn it on, this starts giving water into this bent of tube, and this shifts the bubble back to its original place. So what is the use of reservoir, children? It is used in such a way that the bubbles can be shifted by turning the stopcock on, right? And whenever we don't want, again we turn it off. So once again, the twig is cut and introduced. How I told you, by 2 ways: cutting it obliquely for large surface area and cutting it under water so that no air bubbles are taken up. Then I told you, fill the entire setup with water in such a way that the water is filling the entire tube, and this colored water, the tube is dipping, and we will allow it in such a way that only 1 bubble is introduced at a time. If not, suppose many bubbles are there, we shift it through the reservoir, and the entire setup is ready. Now, what is done further is, as and when the process of transpiration continues from the twig, children, what will happen is a suction force or a pulling force will be set up, and this will pull the bubble forward. So this movement of bubble is noted through the graduated scale, that how much did it move in a given time, and that is converted into the reading that we require. The air bubble can be brought back, as I told you already, by the use of water from the reservoir. That is done. So let us see the working in points. Number 1, transpiration proceeds. Number 2, suction force is set up. 3, water pulled up by the beaker, right? And why this is colored, children? This is specially colored. You can see the pink color, so that the position of air bubble can be noted easily. Because this side it is simple normal water which is transparent, and this side will be colored water, so the easy reading can be maintained. And finally, when water is being pulled up by the beaker, the bubble in capillary tube will be moving. The reading on capillary tube gives the volume of water lost in a given time. And this, children, can be kept in many places. We can keep the apparatus in direct sunlight for a faster transpiration. So if transpiration is fast, what will be the movement of bubble? Yes, it will always be again fast depending on the speed of transpiration. Number 2, if you keep this in a dark room where the transpiration slows down, so this bubble movement will also be slowing down. if it's kept in an open on a cloudy day, again transpiration will be slow, so the movement also will be slow. Alright, then moving further, role of reservoir: to release water into the capillary tube by opening the stopcock, as I already told you, and shifting the air bubble back to its original position. Then there are some precautions to be kept in mind, children. The potometer should be completely watertight and the twig should be cut obliquely underwater. This should not be having any leakage for any drop of water, and also, as I told you, the cutting of twig should be obliquely and done under water so that no air bubble is introduced. Then this method has certain limitations. Let us see what limitations can we discuss. Number one, introduction of this air bubble, though it sounds simple, children, but it's not easy. It's done only by an expert way of handling the apparatus. Number two, the twig may not remain fully alive for a long time, right, depending on the conditions of temperature and weather. And number three, any changes in the outside air temperature may affect the position of air bubble in the capillary tube.